This is the third installment of the Chapter 22 Evidence of Evolution lecture. We've talked about Darwin's finches, we've talked about peppered moths, artificial selection, and even evolution of modern horses. So now we're going to take a look at is the anatomical record and how we can look for evidence of evolution in different fields of biology. Starting with the anatomical record, the theory of evolution really gives us this very logical framework for the understanding of the diversity of life. Many of the observations we make in fields of biology can't be understood in any other way than as the result of evolution. Let's begin with homology. Basically what this is looking at is that the same bones are used in a variety of ways as vertebrates evolved. Consider the idea of homologous structures. This is something that we've definitely taken a look at in regular biology. Here, the idea is that structures with different appearances and functions all derived from the same body parts in a common ancestor. The classic example here is vertebrate forelimbs. All the bones in the vertebrate's forelimbs are the same bones, but over time have been modified or changed in their structure in order to accommodate different means of locomotion. Why should these bones or these limbs be made of the same bones if not due to evolution? Why would the same bones exist? Natural selection simply has modified the structures from a common ancestor for a variety of needs. Consider excuse me, amphibians, reptiles, birds, humans, cats, these being all of your mammals down here, whales, and bats, all containing the same bones, but used for flight, for swimming, for walking on all fours, or for swinging through the trees, if you will, if you look at our ancestors. One example of evolution in terms of homologous structures. Another example is through embryological development. When we consider vertebrate embryos, we see a very similar developmental pattern early on and then differs with time. For example, in vertebrates, in embryos, there are many similarities during embryonic development that we don't see once the organism develops. For example, human and fish embryos both have pharyngeal pouches in the embryos during development. We see gill slits. We see glands and ducts that disappear later on. There are tails in human embryos that develop later on and becomes our coccyx. Human fetuses possess lanugo, which is a very fine fur during the 5th month of embryonic development that goes away. And we see many similarities across the vertebrate embryos that are due to the same genes during development that later get turned off or are not part of development later on that couldn't otherwise be explained other than through the common ancestor. Another example are imperfect structures. Now, what is this? Now, even though natural selection chooses, if you will, the best suited for the environment, not all individuals are ideally suited to their environment because natural selection can only work with the existing variation. Can't just make stuff up. So let's look at this example. Geese and plesiosaurs both had some still do, the geese still do, plesiosaurs are of course extinct, very long flexible necks, many cervical vertebrae, that being your neck. Geese have 25 vertebrae, the plesiosaurs had 76. Giraffes, just like us, have only 7. And in fact, all mammals, just 7. No variation in the vertebrae number, so selection had to work with the size of the vertebrae instead, right? They have really long necks, but they don't have extra vertebrae, they just have really big vertebrae. Another imperfect structure, a very kind of classic example, is the blind spot in our eyes. Now, vertebrates are photoreceptors, the photoreceptor cells that are perceiving our light. They face backwards, or they're more set in the back of the eye, if you will. So this means that the nerve fibers have to exit the back and extend into the eye chamber